Cross4Rare gives access to a comprehensible research opportunity and strategy service. In this program, we can provide you with research status, gap identification, how to attract pharmaceutical company to your disease, and develop your scientific community. We give you ways to support drug development, from target identification to clinical trials, as well as a target scan service. The following case study presents the research status, the gap analysis, and a 6-month strategy proposal for Barth Syndrome Foundation that wants to emphasize its support to drug development. Barth Syndrome Foundation is a non-profit foundation. They work on an ultra-rare disease, and they aim to cure, ideally, find treatments, and improve quality of daily life for patients. First, I gathered the clinical features of Barth syndrome. It's not my intention to explain the syndrome you work with or live with every day, but rather to expose the main issues in Barth syndrome from the patient point of view. After talking to your patients and listening to their testimony at conferences and reviews, it appears to me that the main problems were the cardiomyopathy, the fatigability and exercise intolerance, the neutropenia, and the 3-mg aciduria. When we ask teenagers or young adults what blocked them in their life, they answer heart failure, fatigue, and fear of infection. What I see as a consequence of this tableau is first the challenge for diagnostic and research due to phenotypic diversity, and second, outside of a medical point of view, it's the social aspect of patient alienation. So what has been done up to now for Barth syndrome in terms of academic research? In the center is a map of the federal funding. What is very positive is the effort made for the development of various models, from cellular to animal models. You can see that the mainstream of research has been focused on one of the consequences of the TAS mutation, the cardiolipin dysfunction, and surprisingly, very little has been done on the function itself of the protein of interest, tafazine. Some studies have been also performed on the more general function of mitochondria and on lipids, but really too few on neutropenia and hematopoietic differentiation. In Europe, the research is performed in a different optic, focusing on stem cells in cardiomyopathy, neutropenia, the tafazine involvements in cell differentiation, and most importantly, cancer. The research in China has similar focus as Europe, with the tafazine in cell differentiation and cancer development, as well as cardiomyopathy. Your foundation has changed orientation, putting efforts in gene therapy and starting studies on genotype-phenotype study, as well as neutropenia mechanisms, but still keeping developing models and fatty acid oxidation. That is the global status for research. Now, where are the gaps in research? Gaps in research depend on what you want to obtain: knowledge on the disease, or where and how to act in order to find a cure. We know that the TAS mutation means no functional tafazine that triggers impaired ratio of ml cardiolipin. Well, it's not that simple. Here you see the representation of a cell with the nucleus, where the TAS gene is in the DNA. It goes to the endoplasmic reticulum for transcription and then translation into the protein tafazine. Tafazine has an address that should take it to the mitochondria. Most of the research, as we saw before, has been done on cardiolipin or the mitochondria. But the consequence of the mutation does not stop there. We know that the cytoskeleton, what gives its shape to a cell, is altered in Barth syndrome cells patients. And it has been also shown recently that proteins that are in the rest of the cells are also modified. Through the techniques of proteomics that you might have heard before, they saw that 91 proteins that are not at a normal level in Barth syndrome. Each of these proteins have a function. A protein sometimes needs to be activated in order to exert its function. This state of activation depends on its level of phosphorylation, which means that the protein has a phosphate group attached or not. The general state of phosphorylation is what we call the phosphokinome. A very recent study has shown that the phosphokinome in Barth syndrome is altered in many different functions of the cell. This kind of research will reveal new pathways triggered by the lack of tafazine, and then new targets for drug development to get away from this highly specialized approach and lack of investment in therapies. What are the objectives of your foundation? Improve daily life. I would start with raising awareness in the medical community. I will talk about that later. Cure with gene therapy or find treatments with drug discovery. In both cases, we deal with drug development. But what does that mean, drug development? The drug development ecosystem has different entities interacting together and very dependable from one another. In the middle, I put the technical process of drug discovery, that is pharmacology. This is fed by the academic science generating new ideas and targets. The patient is extremely important, giving information, tissue, and being the beneficiaries. The pharmaceutical industry that has the money and the expertise, and the FDA that has the final decision and is a partner that can be of good help from the beginning. Until recently, the technical process of drug discovery was exclusively owned by pharmaceutical companies in terms of knowledge, expertise, money, and decision-making. For a few years now, the foundations became proactive and involved in pharmacology, pushing or attracting companies to their fields and their disease. The problem is that the knowledge in pharma is kept in pharma, and if you were not trained in the industry, there are no school and academic labs that will give you this expertise. Foundations encounter diverse challenges that a report from the Milken Institute nicely exposed. This is a table from their report. It is noticeable that the key challenges are what makes the daily job in the pharmaceutical industry. Validation and translation study is the starting point of any project, then followed by the drug development in collaboration with medicinal chemist groups. Foundations need the right partner at the right time, but they don't really know who and when. And they have limited knowledge of business strategy and development. There is also the challenge of IP. They don't have easy access to patent attorney as the private sector or university do. I adapted another table from the Milken Institute study that presents the different types of foundation and how they approach the challenges we just talked about. I place your foundation in between the academic support and the supporter. You could cover the 6 challenges, and I can provide you with the medicinal chemist and patent attorney consultant. You can also have access to chemists through the NIH NCATS organization, as I will show you later in the presentation. I added a circle around the last type of foundation, the for-profit, in reference to your wish to support gene therapy. We can talk about it further at the end of the presentation. The strategy proposal I'm going to develop here is in 3 points. Attract pharmaceutical and biotech companies, invest in drug discovery, and I will explain what is drug discovery and what are your opportunities. And the last point, expand the patient population. In order to attract pharmaceutical companies, you need to be able to satisfy a minimum of prerequisites that can be called the de-risking platform. It will convince them that you are prepared for their molecules and clinical trials. They can ask you, where are the patients? You have a patient registry, which is important for the clinical trials, but also for the post-marketing surveillance. What about the patient engagement? You have patient advisors and reviewers. What about the path to approval? FDA endpoints. It will be a global effort with clinicians, patients, and FDA, of course. I will talk about the target product profile and the standardized endpoints for clinical trials, meaning every clinician will measure the same aspect with the same precise methodology. Do you have access to tissue? It would be nice if you had a biobank. I know it's expensive, but you can maybe group with others' foundations to share the expensive. Do you have a variety of targets? Are they validated? That will be the role of your science director, or you can outsource this service. Another question will be, can we test drug easily? You have excellent cellular model and almost the complete panel of animal models. You just need to get good IP agreement to be able to access them. After the animal models, they will ask, are there clinics? There you really need to start working as soon as possible to build a clinic network. Do you have key opinion leaders? Yes, through your conference and workshops. And finally, it would be nice if you had a market model. In the de-risking pharma platform, I will first explain the drug discovery process for small molecules. There are different types of pipeline process. The traditional target-based drug discovery process starts with target identification and validation, where you choose on what you are going to work and be sure that it will improve the condition of the patient. These steps are performed by biologists. Then medical chemists work in collaboration with the biologists to test molecules, improve them, and then test them in animals. When a molecule is proven active, it is also necessary to check it for safety and finally evaluate the toxicity and the efficacy of the drug in humans before reaching the market. This process takes more than 10 years and costs millions of dollars. An alternative to this process is the repurposing drug. In that case, you take a drug that is already on the market, so already went through the process, the whole process. You test it directly in your cellular model or even directly in the animal models. If you see a disease modification, you can directly go in human testing, skipping the toxicology and ADME tests, since the drug is already on the market. It tremendously reduces time and cost, but the drug will be less specific to your disease than the drug that went through the traditional process. A third type of discovery process is the phenotypic high-content screening. It is an efficient strategy when you don't know the mechanisms of actions, but you know the end manifestation of the syndrome and have genetically modified models representing the disease. Let me explain briefly the technique. At the beginning of the development, the pre-embryonic stage, there is just a mass of cells that all look similar. That is, here, for example, a stage of blastocysts, you cannot see an arm or the head. These cells are stem cells, and they will undergo a process of cell differentiation to become more and more different, as, for example, neurons are different from muscles. Recent discoveries in embryonic developments identified the factors that can reverse the differentiation and go back to the stage of stem cells. How is it used? The patient cells contain their genetic material, therefore their mutation. We can induce the cells to become pluripotent stem cells and then differentiate them in the relevant type of cells for your disease. Here is the example of skin cells becoming at the end cardiac cells. Then we can use another technique extremely useful. The cardiomyocyte can be grown in such condition that it will become a 2D functional model, contracting cardiomyocytes, showing all the characteristics of the patient disease. The phenotype of the patient is maintained and can be studied under different drugs or conditions. You can measure the contraction force, the electrophysiology, the metabolic activity, or observe the morphology of the cell. The remarkable part is the high content screening. Because this model is based on cell culture and not animals or tissue, you can have as many cells as you want, so the scale of sample can be really high. The experiments are realized with cells coated on plates the size of your hand that will contain 96 small wells in which you can realize a different condition in each well. So you can test, for example, 96 drugs at the same time. The flowchart is shorter than the two previous drug discovery models. You start from screening, but you can measure several aspects at the same time, including some toxicology. So those effects saves a lot of time. In fact, phenotypic screening has been a dream for years as an alternative to target-based drug discovery that in some cases has its limits, such as poor understanding of the disease and complex causative mechanisms, leading in fact to expensive late-stage failure, which of course challenges financial viability for pharmaceutical industries. Phenotypic screens measure more complex mechanisms, so closer to the disease. They became possible thanks to the iPSC, which is induced pluripotent stem cells for humans, and the genome engineering combining those two, and thanks to big data analysis and small molecule library improvement, and now even more promising with the 3D cell culture and organoid models. Taking back the traditional drug discovery model as a reference, where are the actions that need to be taken in Barth syndrome? First, create a balanced target portfolio. Then define target product profile. Increase repurposing drug testing. Phenotypic screening has to be developed. Then on the animal models point of view, the FDA in fact needs 2 mammal models, including 1 non-rodent, preferentially big mammal. So that's maybe needs to be done for Barth syndrome. Then at the human level, increase patient volume, define FDA endpoints and criteria, create clinic network, and keep improving the natural history. The first step is target identification and validation. Target identification is where you can intervene to repair the broken path. There are 2 levels of intervention. First, tackling the origin of the problem. Here, it is a gene mutation in TAS. The target would be the gene or the gene transcript. Second level is the consequence of the problem. The direct consequence, the protein tafazine does not exert its function in the cell. The target would be the tafazine itself with enzyme replacement. The indirect consequences, what I call the co-workers that don't get their information from tafazine to be activated or deactivated. The target can be any downstream signaling pathway. After having identified the target, you need to validate it. That means checking if modifying the target activity will change the manifestation of the disease. In that case study, Barth syndrome. What are the state of validation here? If you engineer the gene back, the syndrome characteristics are down. The same for the transcript, so the target is validated. The presence of the enzyme, same results. It is validated. For the downstream signaling pathway, the introduction of cardiolipin with nanoparticles failed to restore cell function. That can be due to the technique itself, but the target remains to be validated. And other downstream targets have not been investigated yet. Only the drug ilamipritide has good results, but its exact specificity still remains to be determined. Now that you have a target, you need to find a molecule, a future drug, that is able to activate or block it depending on its function in your disease. Testing a lot of molecules is called screening. You will identify hits that become leads if the hit has a high effect with a low dose or concentration. To perform a screening, you need tools, an assay, which is how to evaluate the action of a molecule on the target, and test system, cellular model or animal models. Barth Syndrome has the luxury to have abundant models. There are different types of source of molecules. You have the traditional drug discovery new molecule entity that we call NME, the phenotypic drug discovery, which will also give new molecules entity, or the repurposing drug that is already a drug. To find a new molecular entity, also called a compound, the screening is performed with chemical libraries. The compounds are incubating with the target. The compound binding or altering the function is a hit. The chemist will find a family of compounds based around the hit, modify them to find and optimize the lead compound for the best efficiency by establishing a structure-activity relationship. There is an NIH affiliated centers that I really recommend. They are specialized in screening with medicinal chemists and all the tools you can dream of. It's named NCATS for National Center for Advancing Translational Science. Having a meeting with them will help to establish the details and plan of screening, taking into account the material and knowledge you have on your disease. You are free to perform your screens in academics labs, but keep in mind that you will not have medicinal chemists and highly specialized pharmacologists, so you will need to seek for this expertise externally. To summarize the opportunity in Barth syndrome target identification, imagine the TAS mutation as a big rock falling from a cliff. You can stop the rock, this would be the gene or mRNA therapy. Or you can let the rock fall and try to stop the other rock it's going to bring along. That would be the downstream signaling pathway, cardiolipin, phosphokinones. What we have now is only symptomatic therapies, improving cardiac efficacy, diuretics, ACE inhibitors, antibiotics, trying to restore the damage of the disease. The action points for Barth syndrome can be gene therapy, going further with the AAV9, the drug repurposing. You already have the ELAM from state therapeutic in clinical trials, but you could increase your participation and join other clinical trials that might benefit your patients, like contact other biotechs like Neurovive for the KL1333 or Reneo Pharma for the REN001 or other biotech companies. For the new targets, the new studies showed a variety of interesting targets and downstream pathways that are of high interest to investigate. I listed here the most relevant to begin with. The phenotypic screening should be one of your main focus, with getting the most valuable chemical libraries from pharmaceutical industry and biotechs. You need to have a balanced portfolio between the risk of failure in development and the benefits your patient will get from the treatment, starting from symptomatic drugs that you have already with low risk but low reward, disease-modifying therapy with repurposing drugs, so you increase the risk a little bit, but the reward is still not really high because they are not very specific to your disease. The new molecular entities are disease-modifying therapies that are at higher risk but highly rewarding, And at the top, of course, the gene therapy. In addition to a balanced portfolio, one of the primary steps to ease the path to pharma and increase the chance of success for treatment is to have a target product profile. This is established with a tight collaboration between the patients, the clinicians, and the help of the FDA. You need to define your goal at two levels: the ideal and the acceptable requirements for the compounds. You need to define primary product indication, the patient populations, the treatment duration, delivery modes, the dosage form, the regimen, the efficacy you want, and establish the risk and the side effect that you will tolerate, and try to find the ideal therapeutic modality. So just to summarize where we are, we have a target portfolio, screening abilities, you selected a hit, then optimized a lead. It works in animal models. Before going to human, the compounds need to be evaluated for toxicology, absorption, and metabolism. The FDA requires usually 2 mammal models, including one non-rodent, preferentially a big animal, like a pig, for example. Here is the status of cell and animal models in Barth syndrome. It's impressive how many models have been developed, really. On the left is the high throughput content with thousands of cells and possibilities to test in the same experiments. At the opposite, on the right side, is a low throughput with one compound in one animal. As I mentioned before, you have human cellular model and the phenotypic model based on iPSC. Then different vertebrate models with non-mammals and mammals rodents, but no model of non-rodent mammal, which could potentially be an issue that you have to check with the FDA to be able to go in human. At the clinical phase stage, the challenge you have are common to most ultra-rare disease. Increase patient population, define endpoint with FDA, and create a clinic network. To enrich patient population, I can see 3 levels of battle. The first one is an indirect way to increase your population by expanding the field and benefit from other disease. Insert Barth syndrome patient as a subgroup in other clinical trials from cardiology, hematology, myopathy, neuromuscular, or others, these others that you have to identify talking with your clinicians. The second way of enriching the patient population is by decreasing misdiagnosis and increase medical awareness. One way is to define the standard of care for each specific medical area in a clinician workshop, for example, and publish them. Another way is having posters and oral presentation in specific conferences where you will reach as many clinicians as possible in the specific areas mentioned before in the expansion of your field. The third level I see is the globalization of your foundation. One big opportunity is the opening of China and its increasing interest in rare disease. If you do the statistics, you will see that there are potentially 3,000 patients in China. That changes your market, considering the 100 you have here. I would contact CORD, which is the counterpart of NORD, the Chinese Organization for Rare Disease. There is another organization, the Chinese Rare Disease Research Consortium. They are relatively new organizations. They have funds for establishing the National Registry for Rare Disease, and they plan to identify 5 to 30 rare diseases genes per year, implement molecular diagnosis and translational research based on these genes, and facilitate the development of therapeutic strategies for rare disease. So that's here, I think, a huge opportunity. In Europe, you can get into Orphanet or ARTOS to create affiliates of your foundations, or get in contact with European Association in the expanding fields. I would start with the AFM, the French Association for Myopathy, but because they already have some interest in Barth syndrome. The first 6 months action point for the research would be to do a target scan, to build a balanced target portfolio with the key opinion leader in a workshop, to start the contract negotiation to access the Barth syndrome models, build a screening library in collaboration with chemists, collaborate with NCATs using the MIP library, explore collaboration opportunities for screens with NCATs or CROs, and start the screen. The action point for the first 6 months on the clinical point of view will be do a clinician workshop to construct the target product profile and also the expansion strategy. Then create a clinic network, identify center of excellence, and start the training. Contact adjacent organizations in cardiomyopathy, neutropenia, and others. Expand the scientific community, attending the Barth Syndrome conference, and connect globally. On the long term, gene therapy should really be in your interest. The science, the AAV9 study performed in the University of Florida is very promising, so you are really close from something that can go to clinical trials. You have anyway to be careful with the overexpression of TAS that leads to tumor. That's something I would really monitor carefully. So the big challenges that everybody will have in gene therapy are the manufacturing and the logistics. There is the demand exceeds the supply. There is only 9 manufacturing organizations. It's growing every day. There are more and more. But that could be, in terms of venture philanthropy, an opportunity for you if you want to go in the manufacturing. The problems are the vectors. The custom process is very expensive and time-consuming. And the vector for clinical trials are 1 or 2 years waiting for production. So there is really here something to work on from the beginning if you want to go to gene therapy. The quality concern is really a problem in terms of stability of the vectors. But the FDA has several initiatives, and you should reach out with them to work with them if you want to increase the manufacturing and the logistics approach for your disease. Maybe you can find something to work with them. As a gene therapy developer, you might want to have the vector manufactured in-house. So that's again the venture philanthropy that you would be interested in. And you would control the VCN, the vector copy number per cell, is also an issue that you can maybe control closely if you are more involved in it. The other big challenge is the logistic, getting the gene and the medicine to the patients. In conclusion, the profile of your foundation with this strategy is in the center of all the stakeholders, being the link between the different entities, the patients, the academia, be a de-risker for the pharmaceutical and biotech company, and be a partner and an influencer on the government and the regulatory and the FDA. Thank you for your attention.